All right, here we are, back from lunch. Thank you all for coming back for this afternoon's really exciting panel we're going to have about exon skipping. To make sure this is a completely unbiased introduction, they invited the gene therapy guy to talk about exon skipping. So here is our crew. We'll go through everyone here in order, and they will have 5 minutes to talk about the approach that they're bringing forward. And to kind of kick it off, some of you might have been in the late transition session. I'm Barry Byrne. I'm a cardiologist by training, a pediatric cardiologist, work at the University of Florida, just up the road. Our focus has been on developing our multidisciplinary clinic for the Southeast. Obviously, some of you who are from the region know our group, and a lot of our research is focused on models, immunology, and gene therapy. But we're here to talk about exon skipping. And so the goal today is we'll just lay a little bit of the groundwork about this technology. There is a little bit of gene therapy associated with this approach that we'll touch on, and then the potential for read-through. So really, how does this technology work? We know this central dogma of where DNA makes messenger RNA, that's what the M stands for, and those messenger RNAs actually carry the instructions to make proteins, all the building blocks of the body. So RNA-mediated therapies don't alter the DNA at all. They just take advantage of the abundance of messenger RNA that exists and make changes transiently in those messenger RNAs to impact the level of protein expression. So it's, of course, important to understand that these therapies are influenced by the mutations you have or your sons have, and then sometimes there's the ability to rejoin parts of the protein that would make it shorter in order to jump over or skip a region where the instructions for making the protein cause it to stop. And those types of mutations are sometimes amenable to repair an effect of the mRNA by bridging one region of the mRNA to another. And that's what you'll hear about today, is the strategies for delivering these molecules, and that's one of the key differentiating factors among the varying approaches, understanding the molecular genetic basis for the processing of the mRNA and ways to improve the efficiency of this process. And you can see here the list of many different exonic mutations that are amenable to this strategy, and the development of them was really influenced by the frequency of any one class of mutations within a region, particularly within the hotspot in Duchenne, which is in and around exon 51. So how does it work? I sort of alluded to the fact that you can piece the parts together in a way that continues the expression of the gene from its beginning to end. So if you think of the codons as words, they are, each codon is made up of 3 letters. Those letters are made up of the 4 bases. And when those are interrupted by a shortened codon or a change in a codon, then the words get mixed up, and that causes either the sentence to stop or an altered sentence to be read through. And if you imagine that when a single word is removed, the sentence might still make sense, but and that would be an example of a mutation that causes Becker muscular dystrophy. When the sentence stops in a very weird spot, like particularly at the very beginning, you have no idea what it was about, and that's not very functional. Sometimes there is natural alterations of that messenger RNA, and these are known as revertant fibers, where the sentence is restored naturally. And it was in that context that this concept was developed as a way to actually restore the so-called reading frame, just like in a sentence in a word. There are obviously many regions where these alterations can be influenced by molecules that bridge these gaps and correct the reading frame. And the frequency of them is kind of shown here. And you see that the most prevalent one was around exon 51, and that's what led to the development of Exondys. So, and now I think luckily with the path forward there was for Exondys and the subsequent therapies that have been developed, we see now the good news is that there are now many sponsors that are working on improving that therapy, addressing its wider and more persistent distribution, and that's where we are today, and we'll hear about that. To contrast this approach to gene therapy, there are a few bullet points here. Certainly, since these are designed for specific mutations, they are mutation specific, and you have to be amenable to that particular product. We've traditionally thought that gene therapies by gene addition are mutation independent. We've learned in the past year, really, that's not entirely true. There are some regions of the expressed protein that are absent in the subject that might be present in the vector, and those can create other immunologic consequences that, for now at least, have excluded those patients, and there are reasonable approaches to be able to get back to that idea that there really is no given mutation that can't be influenced by gene addition. And particularly when an AAV is the means by which an exon skipping product would be delivered, and that is what's also really very recently been demonstrated by Kevin Flanagan's lab at Nationwide. The exon skipping products are given chronically. The frequency is influenced by the mode in which it's given. And when it's given by an AAV vector, it's essentially present continuously, but there is only one of those types of studies underway right now. And theoretically, an AAV vector that would be persistent over the lifespan is a single treatment. And certainly in neurons, that was the goal of the treatment that's been authorized just now over 3 years ago for SMA. Neurons are a bit different than muscle cells. They don't divide after birth. And we know that there's a natural increase in muscle mass over time, and the ongoing inflammation that influences degeneration and regeneration in Duchenne might influence the need for gene therapy that's given more than once. And that's been a focus of what we've been doing. So we think there are possibilities to maintain the level of expression over the lifespan. And these would be very infrequent, but probably more than once would be my prediction. And then, these are, the gene therapy products are not yet approved, and we have now many examples of approved products that rely on the exon skipping technology. So what are the challenges and opportunities of these therapies? Certainly it's getting directly at the cause of Duchenne by targeting a specific mutation. That has some distinct advantages if there are not effects outside of that specific mutation, and that is, I think, a very positive thing about the exon skipping strategies, and that you can then balance between the dose and getting sufficient activity by adjusting in the course of the studies, and then ultimately with the marketed product, the right amount to minimize off-target effects. And it's important to emphasize that in the evolution of this, there's now a lot learned about how to effectively target the striated muscle cells of the heart. So cardiomyocytes are really important, obviously, throughout the course of the disease, but that has to be considered, and I'm sure there'll be questions about that. So they're all different, really, just approaches to restoring dystrophin. We know that is the key interest here. And as I said, this can be done by the various vehicles you'll hear about or in the context of a gene therapy agent. So this was one of the projects that was first discussed publicly at the American Society of Gene Therapy meeting a little over a week ago. I've been following this data closely as a member of the Data Safety Monitoring Board, and there is a very small proportion of Duchenne patients that have a duplication in exon 2, and the skipping strategy here takes advantage of the internal ribosomal entry site, or so-called IRES, that allows for a nearly full-length dystrophin to be made if skipping is achieved by this approach. And so I think one of the important take-homes from the study that was announced last week is that unlike earlier efforts where maybe at the most 50% of muscle fibers might be expressing a functional, even microdystrophin, that they were pretty shocked, I'll say, on behalf of the investigative team, that actually 99% of the fibers in a very young child who received this therapy were positive for dystrophin. And so that, of course, will raise further the question about durability, but it did at least demonstrate that longer dystrophins made by skipping technology, whether it's delivered through any vehicle, can have a dramatic effect as long as enough of the product is getting into the cell to affect that change. And in fact, that was seen in this patient, the third patient in the study, where the CK really dramatically fell to essentially normal after the treatment. And it'll be imperative to really understand how long that will last and what the strategies might be for maintaining that effect. Additionally, there'll be some discussion about read-through agents. This is a strategy to mitigate the effects of a stop codon mutation, which is another form of mutation which can truncate the protein's function. So we'll discuss that as well. And a little schematic about how they work. Again, each time a word is encountered along the sentence that describes the protein, when the word says to stop reading, there is a way to substitute for that and then continue to read through that to get a substitution for that stop to actually make the fully functional protein. And then in terms of understanding, will they work for your son, it's important just to consult with both your genetics team and the neuromuscular team at your site to fully understand whether that mutation is relevant for a product like this. And certainly all the sponsors have worked hard to make sure that there are resources available where you can use the information you have in your own genetic reports to understand eligibility for a study that would use a product like this. So now we'll turn to the company presentations, and I think the first is Gianna from Sarepta. Will talk about the programs at Sarepta. So thank you.
Hello, everybody. It's a privilege to be here with you today, and thank you to Dr. Byrne for setting up such a great conversation around exon skipping and read-through agents. So I will take you through. All right. So here's our forward-looking statements, and for the sake of time, we'll just go on to the next slide. So Sarepta has two different exon skipping platforms in our pipeline. The first one is the PMO technology, which is the basis of our 3 currently approved drugs that Dr. Byrne had mentioned. This is one of the core exon skipping platforms that was led to our first FDA approval back in 2016. We do have 2 late-stage clinical trials that is an opportunity, and I'll speak a little bit about them. But first of all, the Essence study, which is enrolling outside the U.S. for individuals with who are amenable to skipping exon 45 or exon 53. And then I'll talk specifically about the Mission study on the next slide. Our second platform is called PPMO. So you can see by the picture here that we've essentially enhanced our PMO. And what we want to happen with that is if we can get the drug into the muscle cell faster to do its job better, then we get more dystrophin. And so that's the whole aim of kind of redesigning those first generation PMOs. We have started one of our first exon candidates in the PPMO realm. It's exon 51 skipping. We have completed the first part of that trial, and I'll talk to you about the second part of that trial. So back to PMO. Eteplirsen was our first drug that was FDA approved. The FDA approved dose is 30 milligrams per kilogram per week. And we want to see if maybe having a higher dose might give us more dystrophin. I think that's one of the limitations or the things that we can improve upon with first generation drugs is that we need more, we need to get some more dystrophin and see if that changes how things look clinically. We want to change the trajectory of the disease. So for this particular study, it is enrolling in the US as well as outside the US. There is no placebo group, which I think is an important consideration if you're wanting to go into the trial. We're comparing 2 higher doses with our FDA-approved dose of 30 milligram per kilogram. We are looking at ages of 4 to 13. They do have to be ambulatory. It's about 144 weeks in length. These are weekly infusions, so another important consideration for families. There are 2 biopsies that are considered because we've got to take those biopsies at the beginning and then at some point in time after you've been on drug, because we want to see, are we getting higher levels of dystrophin within the muscle? And unfortunately, that's the only way we can look at it right now is to do one of those dreaded biopsies. And then moving over to the PPMO, so we're very excited about this. Like I said, our first exon target was exon 51. We completed part A of this study. We're in part B now. So part A was where we try to figure out what dose is going to look the best. So we balance that between efficacy and safety. So we were able to choose 2 doses to move into part B. Again, this study is enrolling primarily in the United States and Canada right now. There is no placebo group, so you'll be chosen to go into one of those active drug arms. We're looking at a broader range of patients, so ages 7 to 21, ambulatory and non-ambulatory, so we're really happy with that. It takes about 2 years to complete the study, and the great thing is that this is dosed every 4 weeks, so we've been able to make that improvement with this, so I think that is a nice upgrade from where we are with the PMOs. These are I'm so glad that Dr. Burns showed the clinicaltrials.gov website because that is a great place to keep up with where the locations are. This slide was as of April 29th. We have a lot more sites there, but you can go in there and see if there's one that's located closer to you. And just real quickly, we have a lot of ways to stay connected with Sarepta. We would love to connect with the patients and the families, whether it's through our patient affairs and advocacy group. They're here. You can email them at advocacy@sarepta.com. Another great way to keep in touch with our clinical trials is through our Sarepta Ally, and that has the same email address, sareptaally@sarepta.com. These have patient navigators that can help talk more specifically about the trials and line you up with where something might be closer to you. So thank you so much to the patient and families who've been part of our clinical development program, and special thanks to Cure Duchenne for giving us the opportunity to give you an update. Thank you.
Thanks very much, Deanna. And now Jay from NS Pharma is going to talk about their program.
Good afternoon, everyone. As Dr. Burns said, I'm Jay Chauhan from NS Pharma, Medical Science Liaison. So I'm going to just talk about Viltepso. If some of you don't know about our clinical development program, I'll speak a few words on that. And then lastly, I'll show you our pipeline side and what we're working on in the future. Forward-looking statements, we'll just skip over that. So for those of you that are not familiar with NS Pharma, we're based in New Jersey, and our parent company is Nippon Shinyaku, and they're based in Kyoto, Japan. They have over 100 years of experience in the industry. Viltepso is our first commercially approved product here at NS Pharma, and is an antisense oligonucleotide, so an ASO. And it's for patients who have a confirmed mutation of the DMD gene that is amenable to exon 53 skipping. This was approved under the accelerated approval pathway based on increase in dystrophin production, and I'll show you some data shortly. And then our continued approval is contingent on a confirmatory trial, which is a phase 3 trial that is currently ongoing. The dose of Viltepso is 80 milligrams per kilogram, so it is weight-based like similar exon skipping agents, and it's administered once weekly over 1 hour. I know Dr. Byrne had a similar pie chart on the slide. This is just representing Viltepso is for patients amenable to exon 53 skipping, and that reflects about 8% of the Duchenne population. For Viltepso, our clinical development program, the boxes you see in gray are the completed studies, and then the 2 boxes you see in blue are the ongoing studies. If I can direct your attention to the right, our first trial was a phase 2 study here in North America, and that was the trial that allowed us to have accelerated approval. We did complete a phase 2 extension, long-term extension data trial, and that spanned to 216 weeks, almost 4 years. I'm excited also to announce that we have interim data, which covers 2 years, which should be published shortly, and it's open access. Currently ongoing, we have a phase 3 trial, which is going to be double-blind, and it's going to be placebo-controlled, and that's a global study. And lastly, we also have a phase 2 trial with Viltepso currently ongoing for 48 weeks, and this one's going to be looking at patients in an older age group, so at least 8 years of age or older, and we're going to be looking at non-ambulant and ambulant patients. And some secondary outcomes will be looking at pulmonary data as well. So in the original phase 2 trial, which at 25 weeks, we showed mean dystrophin levels increase from 0.6% baseline and to 5.9% average at week 25. And all patients that were in the trial expressed an increase in dystrophin production. We can make dystrophin, but what's the clinical efficacy? So we look at motor function tests. At week 25, these charts represent, the blue lines represent Viltepso, and the gold lines will represent the natural history group that we compare to. You can see in functional test, Viltepso showed improvement or maintenance of function. Some safety parameters. We know exon skipping agents as a class may have some kidney toxicity. However, in our trial, no patients experienced kidney toxicity during treatment of Viltepso. There was no discontinuations or deaths. And all patients that were in the original phase 2 trial continued for the long-term 4-year extension. Common side effects we did see were upper respiratory infection, cough, and fever. And keep in mind these patients were on a stable dose of glucocorticoids, so that's consistent with that. So our commitment to Duchenne doesn't stop here at Viltepso. We have a pipeline in progress. Most recently, we have our compound NS089, and that's to target exon 44, and that's beginning shortly. Other things we're interested in is commercial business development with other companies. We recently had a press release where we partnered with Capricor Therapeutics for their CAP-1002 product. And lastly, we understand that the diagnosis of Duchenne or Becker can be overwhelming. I just want to mention that we have an NS support team and dedicated case managers to help you through the logistics, because the diagnosis can be overwhelming, but getting access to treatment shouldn't be. And I encourage everyone to please visit our booth. Myself, I have Jonathan, Judy, Jules. So anyone with a name starts with J at the booth, please, you'll be in good hands. Thank you.
Thanks very much, Jay. Now Brett from Crawford from BioMarin is going to speak to us. So thanks, Brett, for being here.
Thank you. So in contrast to the previous 2 talks, what I'm going to share with you today is preclinical work. So this is before initiation of human clinical trials. So I'll tell you about BioMarin's what I call 3rd generation exon skipping program for exon 51 skip amenable DMD. It's BMN 351. So our work in this area started really in 2014 when we unsuccessfully tried to champion the approval of a drug called drisapersen for skip amenable DMD. Since that time, we've been focused in the lab to try to identify how to make it better. We've been focusing on 2 attributes, the safety of the drug and then also the extent of skipping so that we could drive dystrophin levels higher. To accomplish that, and I'll tell you about today, we've really focused on 2 different areas. First, the biology. we've gone back and questioned our assumptions and looked for what are the best ways to induce skipping of exon 51. We've also looked to the field of oligonucleotide chemistry and identified improvements in the oligonucleotide structure that can make it a better drug. And then we've leveraged our experience with drisapersen that went into clinical trials and failed, and our learnings in the preclinical models of what it's going to take to succeed, and we applied that to our development of BMN 351. And so today that's led us to this program informed by that experience. So on this slide, I'll tell you about two different aspects of BMN 351. First, the biology, and then the chemistry, and what we've done to make it better. On the top of the slide, the gray bar represents exon 51, from one end to the other, the exon we're trying to skip. And then the vertical bars indicate different oligonucleotides targeting different spots in exon 51 and how much exon skipping they induce. And so the area in the bracket was where drisapersen and all clinically used exon 51 skipping programs target. They all target the same area in that bracket. And so what we found, we stepped back and we designed oligonucleotides going across the entire exon to figure out what's the best way to do this. And what we were surprised to find is that there is a different site that we didn't work on previously that looked like it induced better skipping. So I'll tell you the story in the next slide about how we've worked both to optimize that original site, but then also explore this novel site. And then the novel site ultimately led to BMN 351. The improvements we made in chemistry with BMN 351 includes introducing locked nucleic acids and 5-methylcytosines, which are changes that we think will make the drug higher affinity, more stable, and safer. So this slide shows our work in optimizing drisapersen. So you can see on the very far left of this, this is testing the oligonucleotides in a mouse that has a human DMD gene, and we're measuring the amount of skipping. On the very far left, we can see untreated animals, the vehicle, and then relevant comparators. So drisapersen is probably the most important for us because we know what that did in patients. So we're trying to improve on that. So when we optimized the original site, we had all sorts of oligos, and you can see some of them, oligo H especially, were almost 30 times better at inducing skipping. However, if I adjust the scale here, I'll show you the results from the novel site. So this is the same exact assay tested head-to-head, and we were finding oligonucleotides that were actually over 120-fold better at inducing skipping of exon 51. So we were very excited about that and proceeded forward in its development. So this data is looking at now in a DMD mouse model that's engineered to have the human gene disrupted in a way that it's responsive to exon 51 skipping. So this mouse doesn't have dystrophin, but if you skip exon 51, dystrophin comes on. So on the graph on the left, we're looking at a quadricep, so skeletal muscle, and looking at the induction of dystrophin 1 and 3 months after dosing these animals for 25 weeks. And we can see at the latest time point, there's 2 doses, the low and high dose. The latest time point, we're exceeding 50% of normal dystrophin levels in these mice. And really importantly, as was pointed out earlier, restoring dystrophin expression in the heart is also important. So looking at the heart in the same experiment, we could see we were approaching 30% of normal dystrophin levels. This is far higher than what we were able to obtain with drisapersen. So really importantly to see if that induction of dystrophin translates into function. We then looked at these same animals in what we call the motor rater scoring system. This is looking at the gait and the ambulation of the mice. And we found that in normal DMD mice that are not treated, that gait score increases, indicating the progression of disease. And then in the animals treated that I showed dystrophin levels on the previous slide with the low and high dose of 351, we can prevent that progression in the gait phenotype. So we're very excited about this data. The team right now is really focused on submitting an IND to the FDA. We're aiming to have that submitted in the middle of this year. And if the FDA has positive feedback, then we're hoping to start a study this year. For any other details, up-to-date information on our corporate website under the BioMarin Patient Advocacy tab, you can always find the latest information or reach out to them at the email there. Thank you.
Thanks very much, Brett. Now Shelley from Wave will talk to us. And I'll move over here. Get out of the limelight. Oh, they brought a chair. Too late now.
So at Wave is a biotech company specialized in oligonucleotide, which is a So earlier we had announced a positive result with the molecule designed with stereo pure PN chemistry in CNS disease. And also we proved this chemistry works in DMD. So we designed a molecule for human exon 53 skipping. So here comes the star of my today's presentation. We call this molecule WVE-N531. I know, I hope it has a more cool name in the future. And wouldn't it be nice if we called R2D2? So right now, this molecule is being tested in a clinical trial. The clinical trial has two parts. First part, part A, is a much smaller. We'll dose patients up to 7 doses. And at the end of the study, we'll collect biopsy sample. If the biopsy sample is positive by the end of the year, we'll move forward to part B. Right now, part A is ongoing in UK and Canada. We expect to open US site next year if we decide move to part B. And for both parts, the drug is given every other week. This is less frequent than the once weekly standard of care. Before we can treat patients with this molecule, we have to prove in animals it's safe and it's working. We have collected a big data package for this molecule. And this is just a high-level summary to explain to you why we are so excited about this promising molecule. So in Petri dish, NF31 produced up to 71% dystrophin in patient cells. In animals, it proved to be well-tolerated, safe, and it's working. We also have collected human data, and up to the dose we have tested, it's well-tolerated. And based on both animal data and the patient data, it shows the drug stays in muscle for a long time. And this means we can dose less frequent. So right now it's every other week, and we also hope later we can deliver every month. So conclude my today's presentation, and NF31 is currently being tested in human. We have clinical data and preclinical data show it's safe and well tolerated. Last but not least, I want to express our sincere gratitude to the trial participants, family members, investigators, staffs, and also the whole DMD community. Without your support, we'll never be able to bring a drug to a patient. Thank you all very much. You are the best audience.
Thanks very much, Shelley. And now we're going to hear a little more about how changes in the targeting strategy will help facilitate the delivery of these molecules. So now Mark Stahl from Dyne, from Avidity, is going to talk about their approach. And you'll hear more about Dyne from Ash. So.
Can't confuse Avidity and Dyne. That's just not kosher. That's right. Well, thank you for the introduction, and thanks to Kier Duchenne for allowing me the chance to speak on behalf of Avidity. I'm just going to mention I'm an adult neurologist by training, and I have to say I'm very inspired by getting to see Duchenne kids growing up and making the transition into the adult neurology clinic. It's really an inspiring thing to see, and we're hoping to contribute to that. These are my forward-looking statements, which of course I'm going to read every line of. I'm going to introduce you a little bit to Avidity's technology. What we're doing is bringing an improved method of delivery to an established technology, which is the PMO technology that we've discussed. So our technology is the antibody oligonucleotide conjugate. These are AOCs for short, and we can use them to target muscles. And the idea here, of course, is to treat the underlying genetic cause of DMD. This is how AOCs are constructed. The first part is an antibody. An antibody is something that I'm sure many of you are familiar with, particularly in COVID times. Antibodies are the naturally occurring proteins that we have that fight infections, but we are able to engineer them in the lab to make them bind to almost any protein, including proteins that are on the surface of muscle cells. And that allows us to improve the delivery of something that we attach to it. In this case, we attach an oligonucleotide to it, which in our case is the PMO that causes exon skipping, which we've already talked about at length. So the way that this works is that the antibody itself is able to bind to a protein at the surface of the muscle tissue. And that protein is called the transferrin receptor 1. And what happens when that occurs is that the antibody and the receptor are taken inside the muscle cell, where the AOC is able to release the multiple copies of the oligonucleotide that have been chemically attached to it. And this allows a very powerful delivery of oligonucleotide in terms of its dose, but also its specificity into the muscles. And then, as we've talked about already a few times, the oligonucleotide is able to perform the exon skipping that we discussed, restoring that reading frame and bringing the dystrophin protein back into production. So Avidity is working on several different projects. There are 3 projects that we've been discussing that are exon skipping for Duchenne. The first one that's the closest to reaching clinical trial is AOC 1044 or 1044, for which our first clinical trial is planned by the end of this year. I'm going to tell you a little bit about some of our data, our science. This set of experiments, I think, really nicely illustrates the power of the antibody oligonucleotide conjugate. So here in this experiment, we've taken a single dose at the same amount, so 8 milligrams per kilogram of a PMO, and we've either left it sort of naked or we've attached it to an antibody and given it to an MDX mouse. And what you can see here is that the PMO by itself gives you a detectable but very, very low level of exon skipping, as you can see in the blue. And then in the green, you can see that we're able to cause a more than 50-fold increase in the degree of skipping. So here, at Avidity, we are committed to improving the lives of families whose lives are impacted by rare diseases that we can influence with our RNA-based technology. And so, in order to do that, I think we wanted to show a little bit of additional data that we've just come hot off the presses from the labs and into our hands. So this is some more recent data that shows the distribution of our exon skipping in different tissues, so in calf, in the diaphragm, and in the heart. And then the degree of dystrophin restoration that we're able to get after a single administration of our AOC. And then very importantly, this image, which I find very powerful, is one that shows us that the distribution of that dystrophin is very uniform. And so you want all of the entire muscle, all of its fibers, to be able to pull with essentially the same force. And so you want that dystrophin to be well distributed. And you can see that a targeted distribution of dystrophin through the muscles is able to accomplish that. Sorry. So a few key takeaways from what I've told you. Our AOCs are designed to, of course, treat the underlying genetic cause of DMD in both skeletal muscle and in heart. We were able to see very robust levels of exon skipping, of restoration of dystrophin, and then improvement in muscle function in functional tests that I wasn't able to show because videos take time. We plan for infrequent IV dosing based on the fact that our antibodies last for many days in the body before they're eliminated. As I've mentioned, our first clinical trial in patients is scheduled to begin by the end of this year. And of course, our partnerships with the advocacy groups like Cure Duchenne are critical to us. This is a picture of most of us at a Rare Disease Day event, where I wanted to show a little bit of the faces of the people who work very hard to bring this technology to patients. And so with all of you at the heart of what we do, we want you to feel free to please reach out and contact us at our booth and at our email address, patients@aviditybio.com. Thank you.
Thanks very much, Mark. And now Ash Duger from Dyne Therapeutics will.
is Dyne.
Got it.
Hi, I'm Ash Duger. I'm the head of medical affairs at Dyne Therapeutics. It's great to be here. It's so nice to be here in person with everybody. This is really fantastic. Thank you for being here. Just before I start, I just want to thank Deborah and Paul, the Cure Duchenne team, for inviting us to this great meeting. It's been fantastic. And thank you to the families. It's a privilege to speak with you. So we really appreciate it. So at Dyne, we have a very simple mission. It's to develop life-transforming therapies for serious muscle diseases. That's truly what drives us each and every day. And we try to do our best and work with the community, the physician scientists, and the families to be able to bring therapies that are meaningful to you. Very quickly, there's a snapshot of our pipeline. We have deep expertise in oligonucleotides and neuromuscular space. We have 2 programs that are moving forward right now, one in myotonic dystrophy type 1, which will be dosing in the clinic in the middle of this year, and the other in Duchenne muscular dystrophy for patients who are amenable to exon 51 skipping. As some of you may know, we are on hold. We aren't in the clinic, so there's no untoward issues that we've seen in the clinic. Simply the FDA asked for more information for our application to start our clinical trials, and we are prepared to start our clinical trials in the middle of this year for the 51 skip amenable patient population. Yep. Super. So actually, Mark made my job a lot easier. You're going to hear some of the same themes as I talk about our technology. Similar to what Avidity is doing, we use an antibody linked with a linker to be able to drive our exon skipping PMO into the muscle cells. On the left-hand side of the slide, you can see this Y-shaped figure, a monoclonal antibody. At Dyne, we've taken this top portion here called an antibody fragment or an antigen binding fragment or a Fab that binds to the TFR1 receptor that you just heard about. I'll just add a couple of things. That receptor, it's ubiquitous. It's highly expressed across skeletal muscle, including diaphragm, cardiac muscle, and smooth muscle. And so we're really trying to use the body's sort of natural cell biology by attaching this very sort of small piece of monoclonal antibody that has a high affinity for that TFR1 receptor, and it's attached by this linker, and we attach the PMO with this linker to drive it into the muscle cells. So we're really trying to use this receptor-mediated delivery to drive the PMO into the cell, and hopefully with limited or no off-target effects. Very quickly, our platform has been looked at in the MDX mouse model that you heard about, and we've been able to show very high levels of dystrophin expression, 90% in the diaphragm and 80% in the heart. And to date, other modalities have really just not been able to get there. That's why these next generation exon skipping treatments are so exciting, really being able to not just get into the skeletal muscle, but into these other muscles that are so involved in the morbidity and mortality. And then on the bottom here, we're able to get the dystrophin to the muscle membrane, and that's what you're seeing here. It's important to see that it's localized to the membrane so you have confidence that it's going to have the function that it needs to have. We did test our clinical candidate in larger species in non-human primates, and again, really excited to see the level of exon skipping in the heart and the diaphragm and the quadriceps in these non-human primates. And importantly, we also showed it was safe and well tolerated in non-human primates. So all of this gives us a tremendous amount of confidence as we get into the clinic, hopefully in the middle of this year. Importantly, we have taken a lot of time to listen to the community and the physician scientists. We have designed our program in partnership with the key opinion leaders to be able to have a scientifically and clinically meaningful design that's looking at the right endpoints in the right way and thinking about the natural history so we can interpret our data. And we've also had advisory boards with young men with Duchenne, with parents of young boys, advocacy leaders to give input into our clinical trial protocol, the visit schedule. Where is it going to be most burdensome for the families? What are the main pain points, and what can we do about it? So our design has taken all of that into account, and your input has been directly reflected in our clinical trial design. And very briefly, we are going to have our first in-human study will be a multiple ascending dose study. So we'll be starting and increasing doses along the way. It's placebo-controlled. However, post that placebo-controlled period, we will enroll patients into an open label extension where everybody will receive drug, and it's going to be a global study. Importantly, we're going to be looking at participants who are 4 to 16 years of age, both ambulant and non-ambulant. We know that it's been a struggle to have enough trials in a non-ambulatory patient population, and we really want to make sure that we're serving the full spectrum of participants. We'll be looking at safety, tolerability, dystrophin, and function in this trial. And so we're excited to hopefully start dosing in the middle of this year. Again, this is a picture of the team, and on behalf of Dyne, we thank you all for partnering with us. Thank you.
Thanks very much, Ash. Next up is James MacArthur from PepGen. Welcome.
You got the company right.
Let's make sure you get the right presentation.
Hello, everyone. Good afternoon. Thank you for joining us, and thanks for the invitation to go and discuss the work we're doing here at PepGen. I'm CEO of the company, and we're a Boston-based biotechnology company focused on delivery solutions for therapeutic oligonucleotides. As many of my colleagues have indicated, that's one of the fundamental challenges, is you can have a great oligo, but if it's not getting into the cells that matter, we're not going to be able to impact your children's health. And so that's what we're focused on at PepGen. Here's my disclaimer statement. Now, we're focused on what we believe is a truly innovative solution, a designer peptide that allows us to deliver exon-skipping oligonucleotides for a variety of neuromuscular diseases to the tissues that really matter, not just skeletal muscle, but the diaphragm and also to the heart. We're now a clinical stage company. We've initiated a phase 1 clinical study, which I'll tell you more about, to go and assess both the safety of this drug as well as its ability to go and mediate exon skipping. We also have additional programs moving forward in other neuromuscular diseases like myotonic dystrophy type 1. I'll tell you that in terms of an overview, not only have we developed an approach that we believe allows terrific delivery of the therapeutic oligonucleotide, but it's very safe and well tolerated as well. I'm going to show you just 2 preclinical data slides. Now, we've done a lot of work in mouse models of Duchenne muscular dystrophy and have been able to show great exon skipping, great dystrophin restoration, but unfortunately, the mouse models have not shown to be terribly predictive of efficacy in humans. Non-human primates are thought to be a slightly better model to go and predict what is going to be the efficiency in terms of mediating delivery of the oligo to the tissues that matter and mediating then exon skipping. In this particular case, we're looking with our lead program, EDO51, which mediates exon 51 skipping. Now, one important thing we did in these studies is we actually ran the most clinically advanced PPMO program today, another program that uses a peptide to go and deliver a therapeutic oligonucleotide. This is a molecule that we synthesized, and it's represented in these charts in the purple lines. Our drug, EDO51, is represented in the teal color. I'm not quite sure what to call that color, to be quite honest. But you can go and see when we look at doses of 10 mg per kg or 30 mg per kg that we see superior levels of exon 51 skipping in 2 different skeletal muscle tissue, and in fact in every skeletal muscle tissue that we've looked at today, compared to the more clinically advanced molecule. Indeed, at 30 mg per kg, which we believe will be our target dose, we can go and achieve 73%, 77% exon 51 skipping in these 2 key tissues. Higher levels of exon 51 skipping will lead to higher levels of dystrophin production in patients. When we go and look at the 2 tissues that are driving much of the morbidity of this disease, the heart and the diaphragm, and in particular focusing on the left ventricle and the heart, which is particularly impacted in this terrible disease, we can go and see again terrific levels of exon 51 skipping mediated by the EDO51 drug. This is actually our clinical compound being tested in non-human primates. This, I should mention, in this particular study, is following 3 doses dosed once every 2 weeks in non-human primates. We predict that when we move forward clinically in patients that we'll be looking at a once-a-month dosing regimen, again looking at IV delivery. Again, you can see in the left ventricle that we see really terrific levels of exon 51 skipping following 3 doses compared to the more clinically advanced molecule, highlighting the difference between the teal and the purple here. And similarly in the diaphragm, excellent levels at both 10 mg per kg and 30 mg per kg in the diaphragm in terms of mediating exon 51 skipping. Now, I mentioned that this program is now in clinical studies. We've advanced this program now into a phase 1 healthy normal volunteer study, the outline of which is shown on this particular slide. Now, why are we testing this in healthy volunteers as opposed to patients? We believe that we can glean a tremendous amount of information from this study and hopefully allow us to move into patients with more clinically relevant doses than what one would typically have to do in a first-in-human clinical study. This study is progressing well, and indeed, we plan on, in this study, going to 20 mg per kg. We anticipate being able to go to higher doses based on the toxicology data in both rodents and non-human primates when we ultimately go to patients. This study will yield not only the safety data that will allow us to hopefully start off at higher doses when we begin to move forward with patient studies, which will happen both in the US as well as in Europe, but will also allow us to go and assess what was the efficiency in humans of our ability to go and deliver the therapeutic oligonucleotide to biceps, and what level of exon 51 skipping did that mediate in healthy normal volunteers. This will allow us hopefully to predict what we can do ultimately when we move this program into patients, which we anticipate being in early 2023. We'll report out the data from our phase 1 study at the end of this year in 2022. The patient study, as I'd mentioned, we anticipate commencing in both the US as well as Europe, and we anticipate starting that in the first half of 2023. This will be a multiple ascending dose study, and we'll be reading out not just safety, but also exon 51 skipping, and importantly, dystrophin production in patients. We look forward to coming back to the community later this year and reporting back to you the data from our phase 1 healthy normal volunteer study, and look forward to engaging with you in terms of moving forward with our patient study early next year. Also present at this meeting is Michelle Mellion, our head of clinical development, so feel free to come up after this, either to myself or Michelle, and we're happy to tell you more about this program. Thank you very much.
Thanks very much, James. And Narissa is up next from Entrada.
Great. Hi, everyone. Thank you first to Cure Duchenne for welcoming us, and also to the whole community for welcoming Entrada Therapeutics today. Also, our disclaimer. So at the heart of our work to bring innovative therapies to people living with devastating diseases like Duchenne muscular dystrophy are our partnerships with community leaders and patient advocacy groups. We really want all of our drug development programs to benefit from your wisdom and experience. And one example of how we have already benefited from that wisdom and experience is related to how we chose the first program that would be our lead candidate. So that is ENTR 60144, where we're pursuing a treatment option for patients that are exon 44 skipping amenable. As many of you are aware, and some of the panelists have already spoken to, there is not currently a drug approved for patients that are amenable to exon 44 skipping. So that information coming into us, listening to the community, really helped us to drive that decision making. You've heard from some of the other panelists as well about oligonucleotides, and one of the issues that we see is that these oligonucleotides really need to get into the cells to actually have a really strong effect. 75% of all disease-causing targets, in fact, are in the cell. And so one of the issues that we've all struggled with is being able to get drugs into the cell. And so at Entrada, we have a proprietary technology called EEVs, or endosomal escape vehicles. And we believe that we've really figured out how to get drugs intracellularly. So these endosomal escape vehicles are small cyclic peptides that do two important things. One, they allow for very high intracellular uptake into the cell. And then importantly, they allow for the escape of those oligonucleotides, in the case of Duchenne, to escape from the endosome. Once they've escaped from the endosome, they can then get to the target, which is the nucleus, and allow for this important exon skipping. So here you've seen some similar data from other companies. We actually are utilizing a different mouse model. It's called the D2MDX mouse model. It's a very rigorous model of Duchenne. And here on the top, what you see is exon skipping, not only in skeletal muscle, which the example is tibialis anterior, but also in the heart and diaphragm, very important organs for Duchenne muscular dystrophy. We're seeing very high exon skipping, and I should point out that this is after 4 monthly intravenous doses. On the bottom panel, what you see on the far left is what a healthy muscle cell looks like. So that green fluorescence is dystrophin production. As you look to the next panel, that's vehicle, so really no drug. And then next is oligonucleotide alone, or naked oligonucleotide as it was previously referenced. You can see a little bit of fluorescence, but not all throughout the skeletal muscle. And then on the far right panel is our EEV PMO treated muscle, where again, looks very similar to the wild type. So very exciting data for us. We're really excited with this dystrophin production. But importantly, what does that dystrophin production actually do? So here, in this D2-MDX mouse model, we're actually able to measure some functional outcomes. One of those is creatine kinase. So creatine kinase is released from damaged muscles. And as you can see in the vehicle, so there's lots of damage in that muscle, you're seeing very high levels of CK. Whereas in the green on the far right with our EEV PMO, we're bringing that CK level back to normal. So what that's showing is that muscle damage is being reduced. The next two panels, wire hang and grip strength, we're actually measuring how the mouse muscle works. So as you can see, wild type is in light gray and EEV PMO is in the teal color. So we're getting that functional outcome back close to normal with treatment with our EEV PMO. Now moving to our ENTR 60144 candidate, we're able to test that in the non-human primate. After one single dose at 7 days, you can see exon skipping almost to 100% in some of the skeletal muscles, and then also importantly in the cardiac ventricle. Further, on the right-hand side, what we've done is test to see how long that exon skipping actually lasts after a single IV dose. And so currently, the data are out to day 84, where we are still seeing exon skipping. What does that mean for patients and families? It means we think we have a drug that we may be able to dose every 6 weeks or more. So really could be potentially game-changing from the standpoint of how often that IV drug has to be delivered to patients. We will be filing later in this year regulatory documents that will help us to be able to get clearance to move into clinical trials. We will be doing a first-in-human trial in adult volunteers, and then we will quickly move into multiple ascending dose and phase 2b studies where we're looking for dystrophin as the primary efficacy endpoint. So really excited to be near the clinic and look forward to keeping the Duchenne community updated as we get closer. And finally, a hello from Boston. A thank you to our scientists in Boston and our whole team who helped with the data and this presentation. We do look forward to working with you. Our head of patient advocacy, Reagan Sherman, is here, and we hope you'll come and visit us at our booth number 11. Thank you.
Thanks very much, Narissa. Now, Matt from PTC, you get to hear from again. Thanks, Matt.
Great. Thank you very much, Dr. Byrne. I'm once again honored to be able to have this opportunity to speak with you all today, again, this time about our drug, Adalurin. I feel like while I'm the last speaker today, I'm going to really take us on a journey back to the beginning, to the early days of drug development for Duchenne muscular dystrophy. PTC Therapeutics was founded 24 years ago by our current CEO, Stu Peltz, with the goal of developing innovative therapies for kids and adults with rare disorders. Specifically, the company was founded with a focus on developing therapies for patients with Duchenne muscular dystrophy. In fact, when we did our first clinical trial in 2004, that was the first ever clinical trial done in Duchenne muscular dystrophy. And As is usually the case when you're the first, it's the start of a long road of many learnings, and we're really proud to have been able to make those learnings, and in 2014, Adalurin was approved in Europe, and that was the first ever approved therapy for boys with Duchenne muscular dystrophy, specifically for boys with nonsense mutation Duchenne muscular dystrophy, given that Adalurin, as Dr. Byrne outlined, relies on read-through technology, which I'll talk about in a minute. So it's not an exon skipper, but it's read-through technology that's specific for nonsense mutation Duchenne muscular dystrophy. I've already discussed our Emflaza product earlier today, which is currently in the United States for treatment of all boys 2 years and older. As I mentioned, we remain committed to continuing to make learnings and continuing to work to make Adalurin available to patients everywhere in the world who might benefit from the therapy. Adalurin was the first and only approved dystrophin restoration therapy for nonsense mutation Duchenne muscular dystrophy. As I mentioned, this is read-through technology, which basically means that boys who have a nonsense mutation, during the process of making dystrophin, there's a stop sign. That nonsense mutation puts up a stop sign and stops the production of full-length dystrophin. What Adalurin does is it basically turns that red light, that stop sign, green, go, and allows then for the production of dystrophin by basically programming the cell to ignore that stop sign, keep on moving, and make that dystrophin protein level, which has been key then to not only restoring dystrophin levels, but also to delivering functional benefit. Adalurin is currently approved in over 40 countries in the world. We're currently investigating Adalurin in the United States as part of what we call Study 041, from which we should have results in the very near future. And we really hope to use those results to move forward to gain approval in the United States so we can be able to provide Adalurin to even more boys who might benefit. The safety and effectiveness of Adalurin has been well established in boys with Duchenne muscular dystrophy. We've conducted a number of clinical trials over the course since the first one in 2004, and over 800 patients have participated in these clinical trials and in patient registries, so they've been able to provide us a very robust set of data that demonstrates not only the safety of Adalurin, but also the benefits of Adalurin in terms of functional benefits. And these data on the whole have been shown that Adalurin allows boys with nonsense mutation Duchenne to keep walking for a longer time. It delays the decline in their ability to climb stairs, as well as provides benefit in terms of preserving other aspects of motor function. So basically slowing down that progression from slowing of ambulation and function to loss of ambulation to loss of pulmonary function, and of course ultimately to loss of cardiac function. Now, while we do clinical trials to give us an idea of how a drug may provide benefit in the long term, what we've actually been able to do with our STRIDE registry is collect data to show in the real world what does Adalurin do over the long term. So instead of relying on a short-term clinical trial to say what Adalurin might do, we've been able to, through the STRIDE registry, provide long-term data in the real world of how Adalurin protects boys with Duchenne muscular dystrophy against some of the more serious aspects of the disease. We've been able to show through the STRIDE registry, this long-term real-world evidence registry, that patients with Adalurin treatment can walk for 5.4 years longer than patients in the compared to natural history. That's showing that Adalurin in the real world can slow the decline of ambulatory function and delay loss of ambulation. Importantly, we've also been able to show through the STRIDE registry that treatment with Adalurin is associated with a delay in loss of pulmonary function by 1.8 years when compared with natural history data. So here we're seeing with real-world evidence not only an ability to slow loss of ambulation, but also slow loss of pulmonary function, which are really two of the most important aspects of Duchenne muscular dystrophy pathology. So as I mentioned, we're going to continue our efforts to gain approval in the United States with Adalurin. We're expecting results from our Study 041 in the near future, which we will take to the FDA and see if we can move forward with approval in the United States. But we'll continue all of our efforts to make our therapies available to all patients who might benefit. I want to thank the community, to the patients and the families who participate in our clinical trials. It truly is a community effort. We're proud to have been the pioneers, first in the field of Duchenne muscular dystrophy drug development, and we're so grateful for the partnership of all of the community members, patients, families, foundations, and physicians who've been on this journey with us as we all continue to work together to continue to bring therapies for patients with Duchenne. Thank you.
Thanks so much. We don't have an audience Q&A, but we have some time to touch on a few topics that I think will be of interest to the group. So, and I'll take any volunteers, and maybe I'll pick on you first, Hannah, because what do you think we've learned from the first generation therapies that have really enabled this technology to move forward? Now we have new strategies for improving potency of the molecules and getting wider spread delivery. What's next?
You know, I think as I've listened to everybody and all the new technologies are coming, so what we do in drug development is, you know, your first iteration of a therapy comes to market. It's sometimes not the most perfect therapy, but it's allowing you something. And we continue to learn through that way. And, you know, scientists continue to, you know, figure out ways. You know, you heard so many of us talk about these new ways of getting that drug into the muscle cell. And I think that's what's most exciting about drug therapy, is being able to manipulate things to ultimately get a better result for patients. And so, you know, we've learned how these drugs act in the body. Thankfully, you know, what we know so far in our real world experience, as well as our clinical development program from our part, is that they're very well tolerated. The safety profile is very good. So that allows us to expand on that and then find ways to get the drug into the muscle cell better, because ultimately we want more drug in the muscle cell, more dystrophin to hopefully continue to impact what that looks like clinically.
Yeah, in many ways, like the real estate algorithm, that's location, you all want delivery. You want to say something about that, Ash?
Sure. Yeah. So, I think definitely that one common theme here has been delivery. I think the other, which is critical, right? And you're seeing data from a number of sponsors in cardiac muscle, in diaphragm, which has really, just not been adequately addressed with the first generation. So it's wonderful to see that sort of starting to occur. I think the other theme that has come out here, which is really important, is the duration of action. And so you've seen a lot of non-clinical data, like the slide I showed was a single dose, looking at data a month later. You've seen similar data from the other companies. But being able to develop therapies where we can dose monthly or every 6 weeks or every quarter, that is absolutely, something that we must do, because that burden of weekly dosing, for example, with some current therapies that are on the market, it's great to have the therapies. It's a lot of burden, and so if we can also start to have, longer duration, higher magnitude of skipping and dystrophin production, hopefully we start to really flatten out the decline or maybe even reverse it.
So you want to make a comment?
Yeah, I think the other thing we've learned also is how to study drugs. I think that's really, really important, because getting a drug approved and understanding its benefit is more than just showing dystrophin going up. You actually have to show that it has a functional benefit to patients, and clinical trials are really, really hard to design and do, and I think we all learn from each other on trials that were done before, because we all have to be able to show not only what a drug might do in a mouse or a rat, but what does it do with a person? How are we actually impacting the lives of people with Duchenne? That's the most important, and those learnings are hard. And I think that's something that as we bring more therapies forward, we can leverage the learnings that we have had from the past experiences.
So I think Deanna made a good point about the safety profile of these products, and certainly this is a great opportunity for a whole variety of RNA therapeutics. You wanted to comment?
Yeah, I was just going to add, in addition, to the functional piece, right? The community and many, many organizations and groups, including physicians, that have developed the natural history data for Duchenne as well.
Yeah.
That's another thing, that the fact that we have all of this drug development ongoing, that natural history data has really come from all of you.
So maybe I'll ask Mark to comment on advice you might have to a family that's contemplating starting a novel new product compared to a commercially available product.
Yeah, I think that's a question that is a legitimate difficult struggle for any family, right? The trials offer the potential, of course, for a next generation of therapy that, as we've learned here, has benefits, right, over what we've approved already. But of course, being in a trial has a lot of potential drawbacks as well. And of course, probably the biggest one that everyone worries about is the fact that many of our trials are inevitably placebo-controlled, right? It's very, very difficult, even with the large amount of high-quality natural history data that everyone has provided so generously, to get away entirely from having a group to compare a new drug to, for all of the reasons that we've discussed, both efficacy and safety. We simply don't have the ability to make perfect predictions about how a natural history group that was collected in the past with different other medications, or that didn't maybe collect some of the important data that might be related to your drug, would perform. And so that's a potential risk, I think. And then of course, there's a burden of participating in a study as well, that may involve things like muscle biopsies. As we all know, it's very difficult these days to get away from muscle biopsies, even now. But yet, at the same time, there is that great important thing, not only for your family, for your child, to receive the treatment, but also to push the field forward, right? And that's the one of the greatest gifts that a family and a patient can provide, is to help move things forward and make progress. And so I think it's an individual decision that is really something that is really up to that individual family to really talk through with their doctor and their of course the child themselves, and see what their goals are and what's most important to them. And maybe for right now, it's the the certainty of getting the medication that we know has at least the documented effect, or maybe it's the chance of trying something new and pushing things forward.
Yeah. Well, we were pleased to be able to do the first commercial infusion of Exondys from a boy from Jacksonville here in Gainesville, and we found that after that there was a new challenge of being able to get authorization from payers. How, maybe you all want to comment. We've had a lot of experience of that with the hard work of folks at Sarepta to support patients to make sure they have access. Maybe you can start, and anyone else wants to chime in on how they see the future in that area, because it really, working with payers, establishing that ability to respond to denials from payers is really critical to the patient community. So
it is. I think that's one of the, that's one of the things. It's one thing to, know that you're amenable to treatment, and then now we're going to start the process. So here at Sarepta, we have a wonderful program called Sarepta Assist. We have an entire team of case managers and reimbursement managers that just go to town, ready to help the physicians' offices. That they do have to do some paperwork on their end. We try to help as much as we can. The insurance companies get very picky about, what types of informations and characteristics they want from the patient before they approve it. It can be a long process. Some insurance companies will automatically deny. Then we have to go back and we have to give them data. Even if it's data that's not in our label, we try to update them with all the data. And ultimately, we try to get approval. Fortunately, at Sarepta, if we have a boy who has, their physician and family has gone through this long, drawn-out process, we have a free drug program. So we will not let anybody go without drug regardless of what happens with that insurance. And that's so very important to us.
Anyone else?
I think
comment?
one thing is we're beginning to look at the second generation, third generation products that are seeing higher and higher levels of dystrophin production, hopefully when they're tested clinically in patients, that hopefully that approval process also gets easier once you have not just dystrophin data, but you've also been able to demonstrate outcome measures that indicate, indeed, we've dramatically or significantly improved patient outcomes. So hopefully that will drive things forward as well.
Yeah.
I'd like to add, you know, we have a team of managed care medical people that go and talk. Not your insurance company doesn't understand Duchenne. Yep. And you probably know this from PTC and NS Pharma. We have to educate the payers on what all this means, and you know, they get new employees in, you've got to re-educate a bunch of people, so they don't understand the disease process, and that's some of our job, is to educate the payers so that they do understand it more.
So here's a challenging other point is I imagine you all pay close attention to the regulatory landscape and how the agency really is encouraging sponsors to do adequate and well-controlled studies. But now we have to balance that with the notion that really early treatment might have an even greater benefit. This was observed in, I mentioned the nationwide study that Cure Duchenne helped fund. How do you imagine we might see some of these products come into even younger ages if there is a move towards newborn screening, and certainly when there's an affected sibling, that opportunity exists for early treatment? Do you imagine that there is a path through accelerated approval with compounds like you all are studying? Who wants to speculate on that one?
I mean, I think first and foremost, we have to consider safety, right? So as drug developers, we have to think safety, safety, safety. Efficacy is critical, of course, too. So as we think about getting into younger and younger patients, the onus on safety becomes even higher and higher. And so I think and hope that there is a path for that to happen, but it really is about, you know, showing safety in older populations and then moving slowly and cautiously into younger populations.
Yeah, and I think as we learn more as a community about other meaningful biomarkers that are predictive of clinical outcome, and that will help win the day and help even also to monitor response to therapy or combination therapies.
Yeah, I think you brought up very good points. Given this newborn screening, probably the age, the patient would want to have the treatment early so they can help them. And how the developers can address this is something we need to proactively manage. And maybe we can utilize some like modeling, and especially if you have platform established, you kind of know. Then right now the standard age is 5 years old. Maybe the industry should consider moving forward, include younger boys, and how to have the regulatory path for that. And we need to have a conversation with the regulators. Yeah. That's a very good point.
I mean, CK-based screening is the current technology that's being used, but as access to sequencing data becomes more affordable, you could imagine that just like in cystic fibrosis, the mutational analysis will be the predominant way to identify patients, because that's the most easily accomplished on the blood spot card the way it's currently done. But you face the challenge then of functional outcomes are going to be challenging in that age group. So I think it'll be really helpful to continue to focus on both biomarkers and alternatives, maybe even imaging biomarkers that would be predictive of the clinical outcome.
And also maybe the digital device as well.
Mm-hmm.
Yeah,
many evaluation you have
activity
to have
monitoring. We are. How many people have an Apple Watch?
Oh, yeah.
Everyone's monitoring their activity, so yeah.
We have a little bit of experience in a very young patient population. We did a study from 6 months to the age of 4, and it was primarily just looking at how the drug worked in the body, and to make sure it was safe, since we were dealing with such young infants. The functional scale for that group is very different. You've heard about the NSAA and the 6-minute walk test, which are your traditional endpoints. we have to use a different scale, particularly in those less than 2. But we do hope that this helps our learning as all of our companies go forward. It's been mentioned that we learn from each other. And I think with newborn screening, it is going to really prompt us to look at the safety of our compounds in those really young children.
I think it's clear that for most progressive diseases, everyone will acknowledge, including the FDA, that starting therapy earlier has benefits. So I think it's going to be, a little bit of the burden on us, one, to make sure that we have therapies that will be safer in the younger populations, and then, Barry, as you said, having the appropriate predictive markers so that if we can show changes established in older populations, or even if older as young children, that changes in a biomarker could predict clinical benefit and then take it earlier to measure the biomarker in younger patients. I think the path is clear, and I think the path is there. It's probably just a little bit of work we all have to do to get down that path.
So clearly target selection is somewhat influenced by frequency of mutation. But then, on the notion that no one is left behind, how do you imagine that if there are the potential for class approvals, where if you demonstrate safety with a given modality, that even N-of-1 therapies would be conceivable? Is there a means to go down that road?
I think there's certainly a hope that that's the direction we're going in. There's been a certain reticence from the standpoint of regulatory authorities to go and actually embrace that. We've seen it in the context of a couple of things that we've been doing, but it's certainly our hope as at PepGen, we're looking at developing exon skipping approaches for multiple patient populations. Like many folks, focusing initially on the largest patient populations, but hoping to move into more and more patient populations. We have the ability to go and impact smaller patient groups within the DMD community. It's on us to figure out how to best do that in a fashion that makes sense, both from being expeditious as well as capital efficient for the folks who've invested in our companies.
Yeah. I mean, it certainly makes sense because the safety profile it lends itself to that. Ash?
No, I think it's, just to add on to what James is saying, it's going to be a partnership with the regulators, obviously. And as I'm sure everybody knows, when you get to those rare exons, those rare mutations, you cannot do even the kinds of clinical trials, and we're not even doing traditional clinical trials in this space for 51 or 53 or 45. So those kinds of trials that we are doing for the quote unquote more common mutations, we wouldn't be able to do. So we have to figure out how to leverage the work we do for the clinical programs, and what can we leverage there to be able to give the community, the regulators comfort that we are not going to develop anything that isn't unsafe for the rare mutations, because you can't do clinical trials.
Well, even though most of your molecules are regulated in CDER, Peter Marks has made an important initiative with the Foundation for the NIH to develop bespoke therapies that are so-called customized for really small populations where the conventional, adequate, and well-controlled study can't be done, and you really look for some change versus the natural history. Luckily, we have great natural history data in Duchenne. We do need to probably develop more data in younger pre-symptomatic individuals, and then hopefully with a little bit of a throwdown to see if the gene therapy community can get some bespoke initiatives going, and there's a way forward for even this approach as well. So appreciate you all being here. You want one more comment?
I was going to say, I do think though, that path of getting there does go through generating a lot of high quality data in the populations that we do have access to first, and then we can, hopefully move to that stage later.
Exactly. So we appreciate you all coming to this session, and I hope you all have a great afternoon and the rest of the conference. So thank everyone for being here.
Thank you.